Earth channel. Researchers analyzed the DNA sequences from 240 mammalian species, showing how comparative genomics can shed light on how certain species achieve extraordinary feats and help scientists better understand the functional parts of the human genome. By identifying regions of the genomes that are most conserved across mammalian species, they pinpointed the genetic basis for uncommon mammalian traits, such as the ability to hibernate or detect faint scents from miles away. They also found genetic variants that are more likely to play causal roles in rare and common human diseases. Over the course of 100 million years, mammals have successfully adapted to diverse environments across the globe. The Zoonomia Project, a collaborative scientific effort, has undertaken the task of cataloging the genomic diversity among 240 present-day mammalian species. From the aardvark and African savanna elephant to the yellow-spotted rock hyrax and zebu, DNA sequences have been compared to unravel the genetic intricacies. In a special issue of Science, the Zoonomia team has published several papers demonstrating how comparative genomics can provide insights into the remarkable accomplishments of various species. Additionally, this research helps scientists gain a better understanding of the functional aspects of our own genome and their implications for health and disease. The studies conducted by the Zoonomia researchers have identified specific regions within mammalian genomes that have remained highly conserved over millions of years of evolution. These regions, often represented by single letters of DNA, are presumed to hold significant biological importance. The analysis of DNA samples from over 240 mammalian species has revealed the genetic basis for unique traits such as hibernation and exceptional scent detection. The study also identified species that may be at high risk of extinction, as well as genetic variants that could contribute to rare and common human diseases. The DNA samples were collected by over 50 institutions globally, including the San Diego Wildlife Alliance, which provided genomes from threatened or endangered species. The Zoonomia Project, led by Eleanor Carlson and Kirsten Lindblad, has engaged over 150 individuals across 7 different time zones and has resulted in the largest comparative mammalian genomics resource worldwide. This study showcases the enormous potential for comparative analysis to provide insights into mammalian genomes, despite the challenge posed by the vast size and complexity of the human genome. One of the studies published today, led by co-authors Matthew Christmas, a Uppsala University researcher, and Irene Kaplow, a Carnegie Mellon University postdoctoral researcher, along with Eleanor Carlson, Kirsten Lindblad, and other collaborators, found that a minimum of 10% of the human genome has been preserved across species. Significantly, many of these preserved regions are located outside of protein-coding genes. The study also identified more than 4,500 elements that are almost perfectly conserved among over 98% of the species analyzed. The majority of the conserved regions, which have evolved slower than random variations in the genome, are involved in embryonic development and RNA expression regulation. The regions that have changed more frequently are linked to the way an animal interacts with its environment, such as through immune responses or the development of its skin. In addition, the researchers identified specific parts of the genome that are associated with a few remarkable mammalian traits, such as superior olfaction, exceptional brain size, and the ability to hibernate during the winter. The researchers also discovered that mammals with fewer genetic variations in conserved regions of the genome were more likely to face extinction. Carlson and Lindblad proposed that having even one reference genome per species could assist in identifying at-risk species. However, further investigation is required to refine these techniques, as fewer than 5% of all mammalian species currently have reference genomes. In another study, Carlson, Lindblad, and colleagues used mammalian genomes to study human traits and diseases. They focused on some of the most conserved single-letter genomic regions uncovered in the first paper and compared them to genetic variants that scientists have previously linked to diseases such as cancer using other methods. The team found that their annotations of the genome based on evolutionary conservation revealed more connections between genetic variants and their function than the other methods. They also identified mutations that are likely causal in both rare and common diseases, including cancer, and showed that using conservation in disease studies could make it easier to find genetic changes that increase the risk of disease. The co-first authors of this study were Patrick Sullivan, director of the Center for Psychiatric Genomics at the University of North Carolina Medical School, Chapel Hill, and a professor of psychiatric genetics at Karolinska Institutet in Sweden, Jennifer Meadows, a genetics researcher at Uppsala University in Sweden, and Stephen Guzel, an assistant professor of population and public health sciences at the Keck School of Medicine at the University of Southern California. A third study, co-led by Stephen Riley, an assistant professor of genetics at Yale University, and Pardis Sabeti, an institute member at the Broad, examined more than 10,000 genetic deletions specific to humans using both zoonomia data and experimental analysis, and linked some of them to the function of neurons. Several research papers from the Zoonomia project were released today, covering a range of topics. Among them, the studies revealed that mammalian diversification occurred prior to the mass extinction of dinosaurs, provided a genetic explanation for the survival of a famous sled dog named Balto in Alaska, identified human-specific changes to genome organization, utilized machine learning to find genome regions associated with brain size, examined the evolution of regulatory sequences in the human genome, focused on mobile DNA sequences, found that species with smaller populations historically are at greater risk of extinction today, and compared genes across almost 500 mammalian species. For Carlson, Lindblad, and the researchers who have been sequencing mammalian genomes for zoonomia or its precursor projects since 2005, these studies, and the breadth of questions they answer, are only a fraction of what is possible. Lindblad expressed great enthusiasm for the sequencing of mammalian species, and the novel opportunities it presents for exploring genome evolution and human diseases. She anticipates that this dataset, along with other research initiatives, will facilitate groundbreaking discoveries in the field.